Welcome to Part 2, Lecture 10, on the topic of assimilation of mineral nutrients. This lecture is presented by Dr. Nikki Cooley. This lecture is part of the plant physiology subject, which is a component of the agricultural and viticultural degree programs offered at the institution of NMIT. Please refer to the recommended reading from Tays and Zeiger, Chapter 12, for complementary information. Please ensure that you have watched both Topic 4 on nutrients and Lecture 10, Part 1, before continuing on this lecture. In Part 2 on the lecture of assimilation of mineral nutrients, we will look at specific examples of mineral assimilation in plants. This will include nitrogen, ammonia, sulfite, and cation assimilation. High levels of ammonium in living tissues is toxic to both plants and animals. These high levels result if ammonia is not assimilated. Ammonium dissipates transmembrane photon gradients required for both photosynthetic and respiratory electron transport. The figure on the slide represents the movement of ammonia across a membrane. There are three stages to the transport of ammonia across the membrane. At high pH, ammonium reacts with an OH compounds from water to produce ammonia. Ammonia is membrane permeable and diffuses along its concentration gradient across the membrane. Ammonia reacts with the hydrogen ions to then form ammonium. In contrast to ammonium, plants can store high levels of nitrates or translocate nitrates to form tissue-to-tissue without effect. However, if livestock or humans consume plants high in nitrate, they may suffer from a condition called methemoglobinemia. The liver reduces nitrate to nitrite, which combines with hemoglobin. They may also convert nitrate into nitroamines, potential carcinogens. Some countries limit nitrate content in plant material. In nitrate assimilation, plant roots actively absorb nitrate from the soil solution via several nitrate proton co-transporters. Plants eventually assimilate most nitrate into organic nitrogen compounds. The first step involves the reduction of nitrate to nitrite in the cytosol, transfer of 2 electrons by nitrate reductase. NADH donates the electrons. The formula on the slide shows the chemical process of this reaction. Nitrate reductase is the main molybdenum-containing protein in plant tissues. Nitrate is highly reactive, potentially a toxic ion, transported immediately from the cytosol to chloroplasts in leaves and plastid in roots. Here the enzyme nitrate reductase reduces nitrate to ammonium. This involves the transfer of 6 electrons and is summarized in the equation on the slide. Fd is ferredoxin derived from the photosynthetic electron transport in the chloroplast. A small percentage, ranging from 0.02 to 0.2, of the reduced nitrate is released as nitrous oxide, a greenhouse gas. The figure on your slide is a visual representation of the electron flow through ferredoxin. Light reactions in the photosynthesis produce electrons. These electrons enable ferredoxin to be reduced and then oxidized. The electrons are also used in the nitrate reductase stage. Here the enzyme reduces nitrate to ammonium. In ammonium assimilation, plant cells are able to avoid toxicity by rapidly converting ammonium into amino acids. The enzymes glutamine synthase and glutamate synthase are required in addition to 1 ATP, glutamate and a cofactor such as magnesium, manganese or cobalt. Glutamate and glutamine are formed. The figure on the slide shows the chemical reactions of these processes. In ammonium assimilation, nitrogen from glutamate and glutamine is then incorporated into other amino acids via transamination reactions, catalyzed by aminotransferases. Humans and most animals cannot synthesize certain amino acids. Cysteine, isoleucine, leucine, lysine, methionine, phenylalanine, thymine, tryptamine, valine and arginine. In contrast, plants can synthesize all 20 amino acids common in proteins. The nitrogen-containing amino group derives from the transamination reactions with glutamine or glutamate. The carbon skeleton from amino acids derived from 3-phosphoglycerate and phosphoenol pyruvate or pyruvate from glycolysis or oxoalasis from the acetic acid cycle. The figure on the slide describes the amino acid biosynthetic pathway. Glucose is converted into 3-phosphoglycerate. This is then converted into phosphophenol pyruvate, which is then converted into pyruvate, which enters the citric acid cycle. You can see at the various different stages the different amino acids that can be metabolized. Biological nitrogen fixation. In this form of nitrogen fixation, bacteria can convert atmospheric nitrogen into ammonia. There are some situations where plants have used this ability and have formed associations. The table on the slide 12.2 gives some examples of organisms that can carry out nitrogen fixation. For example, the water fern azole has a symbiotic relationship with the nitrogen-fixing symbiote Anabini azole. This bacteria exists in the root hairs or alongside the root hairs of azole. Azole supplies sugar to the bacteria, while the bacteria fixes the nitrogen for the azole. This system works well and is utilized extensively in rice fields in Eastern Asia. Table 12.2 on the slide gives examples of organisms that can carry out nitrogen fixation. These include cyanobacteria, aerobic bacteria, and anaerobic bacteria. Examples of the nitrogen-fixing genera are presented. The most common type of symbiosis occurs between the members of the family Fabaceae and the soil bacteria called Rhizobia. As nitrogen fixation requires large amounts of energy, the nitrogenase enzymes are found in the site that facilitate the high energy exchange of electrons. Oxygen, a strong electron acceptor, can damage these sites and inactivate nitrogenase, so anaerobic conditions are required. Symbiotic nitrogen-fixing prokaryotes live in nodules, which are special organs of the plant host. In legumes, the bacteria induce the plant to form root nodules. The plant regulates gas permeability in these nodules. The figure on the slide illustrates some of these nodules. These nodules contain an oxygen-binding protein called League hemoglobin, confers a pink colour to nodules. It has a higher affinity for oxygen than human hemoglobin. Rhizobia bind to an emerging root hair in response to chemical attractants sent by the plant. In response to factors produced by the bacteria, the root hair exhibits abnormal curling growth, as seen in the figure on the slide. Rhizobia are released into the epiplast. They penetrate the middle lamellae and a new infection thread is initiated. The infection thread branches until it reaches target cells, where vessels that enclose bacteria cells release bacteria into the cytoplasm. The figure on the slide demonstrates this process. At first, the bacteria continue to divide, and then the surrounding membrane increases in surface area to accommodate this. The bacteria then begin to enlarge and differentiate into nitrogen-fixing endosymbiotic organelles called bacterioids. The symbiotes release ammonium that is rapidly converted into organic forms in the root nodule to avoid toxicity. Amides and uricides are synthesized from ammonia and ultimately released into the xylem for transport to the shoot, where they are converted to ammonium. Now we will move from nitrogen assimilation to the compound sulfur. Sulfur is found in some proteins. It participates in electron transport and is found in the catalytic sites of certain enzymes. Most of the sulfur in plants derives from sulfate, SO4, and it is absorbed via hydrogen ions, symporter, from the soil solution. Sulfate comes predominantly from the weathering of parent rock material. Atmospheric pollution also provides a source of sulfate. For example, burning of fossil fuels derives sulfur dioxide and hydrogen sulfide. Plants can metabolize sulfur dioxide taken up in gaseous form through their stomata. Like sulfur, phosphate is absorbed by the hydrogenized sulfate symporter and incorporated into a variety of compounds such as sugar phosphates, phospholipids and nucleotides. The main entry point of phosphate occurs during the formation of ATP, where inorganic phosphate is added to adenosine diphosphate in mitochondria and chloroplasts. Glycolysis also assimilates phosphates. Cation assimilation allows plants to assimilate both macronutrients and micronutrients. Cation assimilation assimilates macronutrients such as potassium, magnesium and calcium, while the micronutrients, copper, iron, manganese, cobalt, sodium and zinc, are also assimilated during this process. Cations form non-covalent bonds with organic compounds. For example, magnesium bonds with chloroform A. Calcium bonds with polygalacturic acid of cell walls. Iron is essential in iron-sulfur proteins and as a catalyst in redox reactions. Plants obtain iron from the soil as ferric iron, Fe3+, in oxidizers such as FeOH2+, FeOH3 and FeOH4-. However, at neutral pH, ferric iron is highly insoluble. Roots have developed several mechanisms to increase iron solubility. These mechanisms include soil acidification to increase the solubility of ferric iron. Roots generally acidify the soil around them by exclusion of protons during absorption of cations. They also release organic acids such as malic acid and citric acid. Secondly, reduction of ferric iron to the more soluble ferrous form, Fe2+, by an enzyme found in the plasma membranes in roots called the iron chelate reductase. The release of compounds that form stable soluble complexes with iron chelators. Gases produce a special class of iron chelators called sidiophores. This brings us to the end of part 2 on the lecture of assimilation of mineral nutrients. In this lecture, I hope you now have an understanding of nutrient assimilation. I hope you can give detailed examples of nitrogen, ammonia, sulfur, phosphate and cation assimilation. I hope you can understand nitrogen in the environment and the role and the complexities that it plays. We also touched on the components of unsimilated ammonium or nitrate and the impacts that may have on plants and the mechanisms they use to deal with this. Thank you.